Can you hear me, Destiny? Can you hear me, Destiny?
Yeah, I can hear you.
Oh, my sound's muted. Okay, let's try this again. Can you hear me?
Yeah.
Oh, perfect. All right, I'll fix that. All right, let's go ahead and get started. Do you have any questions about anything that we've covered so far?
No, I don't think so.
Okay, so just keep in mind this chapter is about enzymes. And so when we're talking about enzymatic reactions, enzymes, just think of them as workers in a factory, and they have specific jobs. Like one person may put tires on the car and another in the engine, and so that's the way that enzymes are. So if they're reacting with iron, that's all they react with. If they're reacting with insulin, that's all they're reacting with. So they're super specific. What it reacts on are the substrates in the body. And there's all different kinds of ways it can do it. It can do positive and negative charges, hydrogen bonds, ionic bonds. All the weak bonds that we're talking about, enzymes wouldn't want to make strong bonds because you couldn't reverse them. And remember, they need to keep working. So if you made strong bonds, it would be like if you glued your own hands to the car tire when you put it on, you would become part of the car and you could never put another tire on. So strong reactions are out for enzymes. It's weak reactions. And then you can mess with the reactions. Clear this. So here's a classic example of how you can use this knowledge of enzymes and their reactions to increase yields for crops. So back in the day, Monsanto, they made a resistant plant. Well, actually what they did is they created Roundup first. And we kind of talked about this in previous chapters. And so in plants can't make proteins. I mean, they can make the amino acids, but they can't get proteins from their diet. There's no proteins in the soil. So what they do is they make sugars from photosynthesis, and we will cover that in chapter 10. And they have a series of enzymes that just rearrange that. You know, they take nitrogen out of the soil to make the amino part, and they rearrange the sugars, since sugars are carbons and hydrogens and oxygens, and they rearrange them to make amino acids. And so just to make this simple, I'm going to call this enzyme A and B and C and D and E. And they, you know, each of these are workers in a factory, and their job is to rearrange a little bit and a little more until they end up with an amino acid to make a protein. So what Monsanto figured out is that there is a thing called Roundup. It's the scientific name is glyphosate. You don't need to know that, but if you go to the store, you probably have used Roundup before. You look at the label, and this is the ingredient. So glyphosate has a strange shape. It looks like a shape that's a precursor. I'm just going to draw a circle. So it's a precursor to what this enzyme C would work on. You could kind of equate it to maybe a tire that's the same size but a different brand. So maybe it's Goodyear instead of Michelin, but it looks the same, and this enzyme, you know, might not even notice that it's a different brand of tire, and it sticks it on anyway. So when you spray Roundup, it makes this worker occupied with the Roundup, and it blocks this pathway. So you can think about it as an assembly line that shuts down. In the end, what this glyphosate does is it binds up this enzyme so it can't work anymore. And in the end, plants can't make proteins. And if you can't make proteins, then you die. So when you spray Roundup on your plant, you're messing up the enzymes in it so it can't make proteins, and that's what kills it. So Monsanto was like, well, this is great, but what we want to do is figure out a way to make a plant that isn't affected by Roundup. So I want you kind of to think about it this way. So let's say that there's 10 C workers, right, in a factory, and we spray 10 units of Roundup on there. So every one of these workers is preoccupied with Roundup. That means there's no workers left to convert sugars into protein. So what Monsanto, so if you owned a factory and all your workers called in sick, how would you make your product? Any guess? You there? So the way that you would make product is you'd hire more workers. So what Monsanto did is they went back into the DNA and they added the gene for C, and they added into the plants a lot of this C. So maybe each one of these makes 10 more C enzymes. And so now these plants have 20 C enzymes, and if you spray 10 units of Roundup on it, you still have 10 workers left over that can make protein, so the plants don't die. So these plants that have this extra C gene in it, we call Roundup resistant. And this is what has been labeled as GMOs, right? Genetically modified organisms. So a lot of people are afraid of this, but honestly, every plant that you eat has to convert sugars into protein. So every plant has a C enzyme in it. What you're doing is you're just increasing the number of C's. So if you were scared of this enzyme, then you should eat plants because every plant has it. There are ways that they can do this that aren't so similar to the natural biology of the plant. They add salmon genes into things to make them frost resistant because they make sort of an antifreeze. So they're putting that in strawberries and other things. And that's, that's a little more Frankensteinish than, say, Roundup resistant plants. But anyway, so you can increase the amount of enzyme to get the reaction to go faster. Again, enzymes are like workers, so you want more stuff to get made in your factory, or you make, necklaces or whatever, then you hire more workers. And of course, you have to supply the workers with the substrate, which would be beads or tires and sucrose or whatever it is. All right, so we talked a little bit about this before in chapter 3, and you guys know that the most important thing in biology is shape. And if you don't know that by now, you should. And so there are two things that change shape that seriously affect the shape of these enzymes. And remember, the enzyme has an active site in it. So if we change the shape of that active site, then the substrate won't fit in there anymore and it won't react. So the two things that can change shape, you should know by now, are temperature. And this is why it's super dangerous when you get a fever, because your enzyme's active site will change. Like, for example, it's not really an enzyme, but hemoglobin binds oxygen. If it's the receptor that binds oxygen doesn't have the right shape, it won't bind oxygen. And so it would be like holding your breath. So you would die. The other thing that could change is pH, right? And so in a chemistry class, usually I'm in an in-person class and I have, 100 students or whatever, 50 students. And I'll ask them, what happens when you increase the temperature of a reaction? Does it go faster or slower, or does it stay the same? And I'd say 99 out of 100 say, well, it's going to go faster. And that's because they're chemists. If I increase, and as a biologist, if I increase the temperature of a living system, it's not going to make them work better. If I put you in the oven, you're not going to work better. If I change the pH of your blood or your cells, you're not going to work better. And this graph shows this. So here's a couple enzymes. This would be just a normal, typical human enzyme. Remember, enzymes are proteins. And we talked in chapter 5 about how proteins have a specific shape that dictates their function. So the optimal temperature actually provides the optimal shape for this enzyme. And you could probably guess what the best temperature for most enzymes to work in humans would be body temperature, which is around 37 Celsius. It's hard to draw a straight line there, but that's what I meant to do. But that's different. So the optimal temperature, or we could say shape, for an enzyme of a bacteria that lives in the hot springs in Yellowstone would be much different. And these can be the exact same enzyme. So for example, if we're talking about, let's just say amylase. That's the enzyme that breaks down amylose. And we talked about that in chapter 5, and that's long chains of glucose. And we need to break that down to release the sugars that we use for energy to make ATP so that we can live. And all living things have amylase. Well, maybe not all, but 99.999%. And so a bacteria would have amylase to break down sugars it encounters in a hot spring. And so it has evolved a shape. Let's just say amylase's shape is rectangular. So the shape amylase's shape doesn't change. It's a rigid, I'm sorry, not amylase, amylose substrate. That sugar doesn't change. Its shape is always going to be this, let's just say a rectangle, or it looks more like a square, but not meant to be a rectangle. And this is the active site of the enzyme. So it has a shape that's going to fit that substrate. And so if I heated up this human enzyme to this temperature of, you know, around 80 degrees Celsius, which is pretty hot, close to boiling, it would change its shape. And so this enzyme from a human wouldn't work under the conditions of a hot spring. That makes sense. If I throw you in a hot spring, you're probably not going to do very well for very long. But it's the reverse is true. If I take the enzyme amylase from a bacteria in a hot spring and I put it in a human, it's not going to have the right shape because it's a different temperature. So you can't really cross-use these enzymes in different conditions. And so every enzyme is going to have what we call the optimal temperature. It's the temperature it works best at. And usually that's the temperature that that organism lives at. So for example, a fungus that's found in the forest floor would have a much cooler optimal temperature for amylase than the one that's found in a human mouth versus one that's found in a bacteria that's found in a hot spring in Yellowstone. And then even if we look at the same organism, so pepsin is something that breaks down proteins. It's found in your stomach. That you probably the word comes from the Greek, which has to do with stomach. So like pepsid, AC, that's like the same word we're talking about. So this breaks down proteins in your stomach. The pH of your stomach is around 2, and we talked about this in chapter 3. And you also have another protein that breaks down proteins in your mouth called trypsin. This is the same sort of stuff that they use in meat tenderizer. So if you get meat tenderizer, take a look, it probably has trypsin in it. And of course, that's going to be at a different pH because your mouth isn't as acidic as your stomach. And so trypsin couldn't work in your stomach, it's too acidic. And pepsin couldn't work in your mouth, it's too basic. They have different shapes. So the moral of this whole thing is that every enzyme has an optimal temperature, the one it works best at. And so on the test, I might ask you, well, this fungus lives at 72 degrees Fahrenheit. What's its optimal temperature for its enzyme amylase? And what would your answer be? Should be 72. And each enzyme has its own optimal pH. So like you wouldn't use an enzyme from the lysosome that's 5 for something in the cell that's 7.2 because they wouldn't work together. A medical doctor that I worked with, I won't name them, they work in the VA now. He was doing experiments on a enzyme that chops up DNA, which is found in the lysosome. And he couldn't figure out why that enzyme wouldn't chop up the DNA. And it turns out that he was using 7.2 as the pH. So I asked him what your pH was. And that wouldn't work because that enzyme's optimal pH is 5. So he had to change the pH, and then the enzyme worked, and then he got his PhD from something that's pretty basic that he should have learned in enterobiology. So anyway, optimal temperature, optimal pH. If you reduce the temperature, the enzymes don't work as well. If you increase the temperature, the enzymes don't work as well. And that's why we have these curves. Same thing with pH. Any questions? All right. Okay, so there are other things that affect enzyme activity. There are things called cofactors. Cofactors are inorganic. So what does inorganic mean? It means it has no carbon. And so these are non-protein molecules because you guys know proteins have carbons in them. So cofactors that don't involve carbon, an example would be zinc or copper or magnesium, iron, that sort of stuff. And so what cofactors do is they bind to, remember when we bind one molecule to another molecule, what does it do to it?
Changes the shape.
Yeah. And so this enzyme might not have the right shape unless it binds its cofactor, which changes its shape. So now that it's on, right? So without the cofactor, there's no cofactor attached to it. It has a different shape and it would be off. So your body can control whether your enzymes are on or off by the presence of cofactors. It's really a smart way to do it because it takes a lot of time to go from to DNA to RNA to protein, right? So if you want to make a protein, an enzyme from scratch, it takes a lot longer than if it's already sitting around and you just have an on-off switch that you can use. Does that make sense? So that's the purpose of these. And a lot of these are things that you would find in, a vitamin, a multivitamin. These are really important. Without these, your proteins wouldn't be able to switch, your enzymes wouldn't be able to switch on and off. These are what we call trace elements, right? Because they're inorganic. And then so most vitamins were organic, like vitamin B, and that means they have carbon in them, right? And so we call them coenzymes. It's just a word you need to learn. So learn coenzymes and learn cofactors and be able to distinguish between the two. So coenzymes are cofactors, but they're not inorganic, they're organic. And they work the same way. They bind to enzymes and turn them on or off by changing their shape. So, diseases like scurvy and stuff like that is caused by the lack of the ability to turn enzymes on or off because you don't have the proper vitamins or the proper dose of vitamins. You can have inhibitors of enzymes, and there's two forms of that. One is called competitive and one is called non-competitive. So the difference is, here's a competitive inhibitor. So this is the active site, the part of the enzyme that causes the reaction to occur. And this would be the substrate here in blue, the thing that it worked on. So we could say that this enzyme is sucrase, right? And you guys know that breaks down sucrose. And so, but if something were to have a similar shape and fit into the active site and block it, then this enzyme would no longer work. It's kind of like if someone parked in your employee of the month spot, you can't park there, right? Until they move. So competitive inhibitors directly block the space of the active site so that the substrate can't park there and the reaction won't occur. You can also have things called non-competitive inhibitors. It's because, remember, if we bind one molecule to another molecule, no matter where it is, what does it do to it? It changes its shape. Yeah, so it could change the shape of this active site in such a way where the substrate no longer fits. So a non-competitive inhibitor binds somewhere away from the active site, but the results are the same. It stops the enzyme from working. And this could be anything, right? This could be like a painkiller, so it blocks a pain receptor, or, you know, most drugs work this way, either by competitive or non-competitive inhibition. Okay, so when we talk about this sort of enzyme control to turn the active site on or off, we call that allosteric regulation. So that's just a fancy word that you need to learn. But basically, it's just regulating whether the active site is open or closed. If it's closed, obviously there's no reaction. If it's open, there would be a reaction. An allosteric site can be somewhere off of the active site, and it can control whether this is on or off. So I'll show you an example of this. So here's a molecule that we call an activator. So this, sort of imagine that this is shut down like this, unless the activator is present. The activator binds, right, to some specific place not in the active site and opens up the enzyme. So in this case, there's 4 different enzymes linked together, and that can happen. It has a quaternary structure, which we talked about in chapter 5. That means more than one comes together to make a functional protein, in this case, an enzyme. And then we can have something called an inhibitor. So the active site of the enzyme would be open until the inhibitor is bound to it, and then it's closed. So we control the on-off switch of the enzymes by either an activator or an inhibitor. And that's kind of cool. And we need that to be able to respond to our environment, which is what we talked about in chapter 1. So I think I've used this example before, but let's say you went outside and you were not properly dressed and it's really cold. In order for you to warm up, your body's natural response is to start shivering. And that has to do with turning on genes and pathways that cause you to shiver. Now, if there was no way to regulate that, when you got warm again, it might take a long time for you to stop shivering. But because we can turn these on and off relatively quickly without having to make them from scratch, then you can respond much better to your environment. So if it's really cold, you start shivering. If you warm up, you stop shivering. And it's almost instantaneous. And that's what we want. That's important to life. All right. Cooperativity is just another term of a substrate that binds, in this case, the active site, that causes the other parts of the, so remember, this is a 4-part enzyme. It has 4 active sites. The other active sites to open, and that's okay. This one is sort of useless at the point where the substrate's in it, but these are, and remember, in an enzyme, it does a reaction and then it can do another one and another one. So this part of this enzyme, this active site, could do maybe 1,000 reactions before it succumbed to that second law of thermodynamics that says that all things tend to break down, right? They lead towards disorder. Like a new car, it doesn't get better with age. So that's what we're talking about, cooperativity. That's how it's different from allosteric, because allosteric is away from the active site, right? Whereas cooperativity is in the active site. Any questions? All right, so it's basically just a trick to change the shape of the active site to turn the enzyme on and off. And so here's some examples of what we might need that. So there are a whole series of enzymes that are designed by your body to break down lactose, the sugar in milk. And making those proteins takes a lot of energy, right? It's like, so let's put it this way. If you didn't drink milk, if there was no milk in your system, would you need the enzymes to break down milk?
No.
Yeah. And you wouldn't want to waste your time doing that either. It would be like, well, if you're on Mars and there's no cell towers, why would you waste your time building a cell phone? Your body is the same way. So it's not going to make the enzymes to break down milk unless milk is present. So what happens is that when you drink milk, the enzyme, so I'm just going to, it's a fake enzyme, but it's called beta-galactosidase. We call it beta-gal for short. And it breaks the bonds in lactose to, lactose is actually glucose, which is the sugar you're familiar with, and another sugar called galactose. And so it's a disaccharide. And this enzyme's job is to break this bond here. And it does it through what kind of reaction? I'll take you back on a trip to chapter 5. When we want to break a bond, what's the reaction? I'll give you a hint. It's either dehydration or hydrolysis.
Hydrolysis.
Yes. So it's going to add water in here, breaks that bond. That's its job. So, but this enzyme isn't going to be active. It's going to be off unless this together is called lactose. It's going to be off unless lactose is present. So what the trick is that it actually uses lactose to bind to it, right? To cause it to turn, changes its shape. That's my futile attempt at the Pentagon. And so that causes it to turn on and become active. And so that's what we call feedback. And it can be positive feedback or negative feedback. In this case, we're talking about inhibition. So here's another thing. So since amino acids are super similar to each other, we have the amino group and the carbon and the carboxylic acid and the hydrogen and the R group. So this is really what makes it different. And maybe it's an OH and you want to make it an H. So it's real easy to turn one amino acid like threonine into another amino acid like isoleucine. Now, I don't care if you guys know these names, but it's basically just manipulating this R group. So maybe we add an oxygen or whatever. And enzymes do that in a pathway. And so let's say that you don't have any isoleucine, but you have plenty of threonine. So you could convert threonine into isoleucine, but you only want to do that if you're low on isoleucine, right? So what happens is threonine binds to the enzyme. It's called deaminase, so it removes an amino group. You don't need to know that, but anyway, so there's an intermediate that goes to enzyme 2 and another one, and enzyme 3 works on that, and so on, kind of like the glyphosate pathway. And in the end, you make isoleucine that your body can use. Now, if you get a lot of isoleucine made, then you want to shut this pathway down because you don't need to make more. So in this way, we have negative feedback. Isoleucine binds in an allosteric way to the enzyme that's responsible for converting threonine into isoleucine. So as isoleucine increases, more of it binds to the enzyme, shuts down its active site, and turns off this pathway so that we don't make any more isoleucine from threonine. And that's what we mean by feedback inhibition. That if you have enough of something, you want to shut down the pathway that makes it. There's another one we'll talk about later called tryptophan. If you ate a bunch of tryptophan, like let's say you had a giant turkey dinner, would you need to make more tryptophan?
No.
No. And so you want to shut down the pathway that makes tryptophan. It's basically how this works, which is quite the opposite of lactose, right? If you have lactose present, you want to break it down. In this case, you have tryptophan present, you want to turn it off. And so your body, there's lots of pathways and stuff that your body does. And this is how you're able to respond to your environment and maximize your energy retention, not waste energy. Okay, so that's the end of chapter 8. Do you guys have any questions about chapter 8?
No, sir.
Okay, so I'm gonna stop this share, and then I'm gonna go to chapter 9. This is kind of a long chapter, and I like this chapter because it sort of tries to tie in everything that you guys have learned so far. So you've learned a bunch of stuff. You're probably like, why the hell do I need to know this? Well, this is why. Okay, so everybody can see chapter 9?
Yes.
Perfect. Let me try this again. Okay, so you should be able to see it now. All right, so we have talked about nucleotides, and we've talked about ATP, and we talked about what we do to, you know, the mitochondria makes ATP, and that's the reason that we breathe air, oxygen, and we eat food is simply to make ATP. That's our energy currency. We've talked about how ATP is such a magic molecule because we can add phosphates to stuff and adding a molecule to another molecule changes its shape. So, you know, we can add a phosphate to allow transport against the gradient. Remember, that's called active transport. We can add a phosphate to a motor protein, like this might be myosin. I know it's tubulin, but muscles are probably more interesting. So we add a phosphate to the motor protein and it allows it to move, right? And we talked about you do the same thing by just walking. That's done on a real small scale inside your body. That's how you lift a cup of coffee. And then we can also use phosphates to change the shape to weaken the bonds, right? The delta G. We can change the energy, the enthalpy, to reduce it by bending the bonds, just like we would bend a stick to break it. That lowers delta G. And we can get reactions that occur in the body that wouldn't normally occur without changing temperature or disorder, which is bad. And so all of these things we can do because ATP, we just add a phosphate and we get all the magic to happen. And then ATP becomes ADP, the diphosphate. Remember, ATP is more like a compressed spring, and we can use it to do work like shoot a ball or BB or whatever. And then ADP is more like an uncompressed spring. So this is ATP and this is ADP. And we put a ball on top of there, and it's not going to do anything interesting. It's just going to sit there. So this has energy, and this has no energy. But we can make this have energy by simply compressing the spring, by adding another phosphate to it, and then we get ATP. So in this chapter, we're going to talk about how we add phosphates to ADP to make ATP. And in doing so, you're going to figure out why you breathe in oxygen, why you exhale carbon dioxide, why you eat food, why certain foods you can't eat dirt to make energy, and so on and so forth. All right, so you probably know this already. It's not a big secret. You probably learned this when you were a kid. Almost all the energy on the planet comes from sunlight, right? And energy and light we learned in here is it travels from the sun, so it's kinetic. And we talked about we can convert kinetic into potential, and during photosynthesis, plants take in carbon dioxide and water. This isn't a balanced equation, and they can only make sugars, but we talked about plants can, use those as a carbon basis to make proteins and DNA and RNA and fats, right? They need to do that so they can not freeze to death in the winter. And plants also have mitochondria, and they use that to make ATP, which does most of the cell work. And we just talked about how it can do that. And there's always, remember, anytime we do that reaction, we tend towards disorder, entropy, and we can't stop it. It's part of that law of thermodynamics. And usually entropy is lost as heat energy, because heat energy is a random form of energy. It just dissipates out into the universe, and you can recapture it, and that's what they do with hybrid vehicles, with regenerative braking, because when you brake, it causes heat, and that heat can be converted into electricity, which is potential energy. We talked about it. But heat energy is kinetic energy. And so, we can utilize this heat to maintain a relatively constant internal environment like warm-blooded organisms do. But you have to do a lot of ATP to be warm-blooded. So you got to burn a lot of energy. All right, so basically we're taking light energy from the sun, we're converting that into the chemical energy of ATP that can be used to do things. And so there's a bunch of enzymes involved in this, and that's why you had to learn about enzymes. And then there's waste products like carbon dioxide. These have their energy poor molecules, right? And then we're storing our energy in the form of ATP, which is a chemical. And when we store chemical energy, even if it's in like a cup of sugar that has calories in it, it's still potential energy. Same thing with electricity. And we use that to perform cellular work. And there's always going to be some heat generated when you do these reactions. So we're going to focus on two pathways here, and there's lots of different things that you can use for energy. You know this, but since this course is not 2 years long, we're just going to look at glucose solely. But just keep in mind, you can use lactose, galactose, proteins, different kinds of proteins, different amino acids to do this. And so we're going to talk about cellular respiration, and we're going to talk about fermentation. Fermentation is no oxygen. Cellular respiration is with oxygen. You can make ATP through fermentation, but you can only make 2 from a mole of glucose, where in cellular respiration, and specifically in humans, you can make 36. So you can make roughly 18 times the amount of energy by breathing as you'd make without breathing. So that's why most people breathe or they die. All right, any questions about anything so far?
This is going back, sorry, I did have one question regarding last chapter.
Sure.
So for the allosteric sites, is it that non-substrate molecules can bond to these sites and change the shape of the enzyme and inactivate the enzyme?
Yeah.
Okay, I just need a clarification on that, but that was my question for last chapter.
All right, let me pull this up. Okay, say that again now. You're saying that a non-substrate.
So on an allosteric site, non-substrate molecule can bond to these sites, right?
Yeah, so that's what it's showing here. A substrate molecule always binds in the active site. So that would be right here. In allosterics, it's another molecule that has nothing to do with the molecule it's reacting with. So, for example, I mean, it could be a substrate molecule as well, but that's what we would call that cooperativity, like here, as opposed to allosteric, which is here. Does that make sense?
Okay, so allosteric is not a substrate molecule, correct?
Right. It would bind somewhere away from the active site. So when we talk about substrates, it's that's what binds into the active site that does the reaction. So here, this would be the active site right here and here and here and here. And so that this molecule binds away from the active site.
And when it binds, it turns off the enzyme.
Well, it changes its shape, so the active site is no, but that's for an inhibitor. So there's two things that could happen here. You could have an activator that turns it on. All right, so let's I'm not going to draw this whole thing out, but let's just say here's our substrate. It's a box. And that substrate is going to fit into the active site of this enzyme. So the substrate would go into this active site normally, and the reaction would occur. Now, you could have something bind to it. And when it did that, it might change its shape. So the substrate no longer fits.
And that would be an inhibitor, correct?
That would be an inhibitor, so it changes shape. Now, you could also have an enzyme that's like this. Same deal, I'm just redrawing it, right? So without anything attached to it, the substrate doesn't fit. But you could have an activator that changed its shape to be square, and now the substrate fits. So that's this here. This is an example of that, and then this would be an example of that. And both are valid. So you can have an enzyme sitting in your cell, in your cytoplasm, that's off, and it's waiting for something to turn it on. Like lactose, for example. So it's off. The enzymes that break down lactose are off, like this one here. And then, so the substrate doesn't fit, lactose doesn't fit. metabolite of lactose binds here and then turns it on. So now it's going to break down lactose. Does that make sense? Which would be an example of an activator. And then tryptophan would be more like an inhibitor. So if you have lots of tryptophan in your cell, you don't need to make more tryptophan. So you would want to, it would be on unless there's a lot of tryptophan, which would bind to it and turn it off. Does that make sense?
So certain, so depending on the enzyme, it'll help help the molecule bond or change the shape for it to bond or not bond.
Right. So lots of molecules can either be inhibitors or activators. And it really depends. I mean, this is a generalization because you literally have 30,000 proteins in your body that have different functions and are controlled different ways. But yeah, this is generally how it works.
Thank you, Professor.
No problem. All right. Any other questions before we switch back to chapter 9 on chapter 8? Okay. So let's see. All right. So we left off talking about fermentation. Remember I told you that we make a small amount of ATP to ATP. Fermentation makes ATP, but it's not very efficient. And you're going to see that this has to do with the transfer of electrons and organic compounds. Remember, organic compound isn't a scary word. It just means it has carbon in it. Fermentation is anaerobic. So remember, ana is not an aerobic. So aerobic means in the presence of oxygen, doesn't require oxygen. So an example of this would be like, let's say that you decided that you were going to go do a cardio workout. So you get on the treadmill and you're running your ass off. This would be the same sort of thing that like a bicyclist would do in the Tour de France. So, you know, you have a certain amount of oxygen that you're breathing into your body. That oxygen is getting converted into ATP. So when you get on the treadmill and you start running faster and faster, what's the first thing your body does?
Starts burning energy.
Well, no, I mean, just tell me, like, what would you say you are? Like, what are you doing when you're running on a treadmill? Are you sweating more?
Oh yeah,
And
heating
you're
up.
breathing heavier. So you're burning more ATP, which increases heat. And we just talked about that, which is you're sweating to cool you down. And you're also breathing heavier because your body needs oxygen to make more ATP. But your lungs are only so big and you can only breathe so fast. So if you kept running faster and faster and faster, you would try to breathe harder and harder and harder, but you couldn't do it. And then at some point, you're still going to need energy to try to get away from that lion or tiger or bear. And so your body is going to go into a fermentation state. This is kind of how Lance Armstrong got away with sort of cheating the system in the Tour de France. So Lance Armstrong has unusually large lungs if you compare to other human beings. They're big, which means he can load a lot more oxygen in his blood. But what he was doing, which, you know, that he wasn't taking illegal drugs or anything. What he was doing is he was blood doping, which means basically he would take blood out of himself the night before. So he'd remove his blood, maybe a pint or two. And then right before the race, he'd put the blood back in, right? And what that does is he has extra red blood cells that can load more oxygen because he has more hemoglobin. And so he can go further before having to go into fermentation. Or remember, once you hit fermentation wall, you're making 18 times less ATP. So you have an 18 times advantage if you can stay in respiration as opposed to fermentation. And that's what he was doing to win all of these races. Okay, so respiration occurs in the presence of oxygen, and it's basically you're making ATP, a lot more ATP than you would with fermentation. And like I said, we're just going to focus on glucose, but you could use anything. You could use proteins, amino acids, fats, other stuff. And that would be more for a biochemistry class than this class. We're just going to focus on glucose. So you take in glucose, you breathe in oxygen, and you exhale carbon dioxide, and a byproduct of that is water, and you're going to make energy as ATP. Remember, we talked about the first law of thermodynamics. It says that energy can't be created or destroyed. So if we're producing energy out here, that means that these molecules have to be higher energy than these molecules. Make sense? To get energy out. So like I said, there's all kinds of other stuff that you can use, but we're just going to focus on glucose. This is the balanced equation. Remember, we have to balance the equation. All right, so in order to understand how this works, you have to understand a thing called oxidation reduction. It's a fancy word, but what it means is that we're oxidizing and reducing molecules in the same reaction. We call that redox for short, which is backwards of what this says, but I didn't make this up. Somebody else did. So you just have to live with it. So what happens is that when we say something's oxidized, it's losing an electron. So lose an electron, you're oxidized. If we say you're reduced, then that means you're gaining an electron. And that makes sense, right? Because you're going to be more negative. So gain an electron, you're reduced. So here's an example. Let's say that this molecule, I'm going to show you real life ones in a second, but let's say molecule A, this could be glucose, for example, is going to give up an electron to molecule B. And this molecule could be, well, let's just say it's ATP. It's not, there's an intermediate, but let's just say it. So if we're taking an electron off glucose and put it on ATP, then that's an oxidation reduction reaction. And so since A gave up the electron, it lost the electron, we say that it is oxidized now. And that means it has a more positive charge because it gave up a negative charge. And B is now reduced, which means it has a more negative charge because it's gained an electron. So if you lose an electron, you're oxidized. And if you gain an electron, you're reduced. And, you know, this is how I learned it. Leo, the lion, goes grr. So lose an electron, you're oxidized. Gain an electron, you're reduced. But you can remember it however you want. You just need to keep this straight because this is really important in biological systems. And I'm going to show you why now. So I don't want you to focus on all this crazy chemical stuff. I know that this can be overwhelming. So just what I want you to see here is that this is a friend of yours. It's called ribose. We learned about it in chapter 5. We know that because it has the hydroxyl groups there on the number 2 and the number 3 carbon. Adenine is a base, which you learned about in chapter 3. It's the A, right? Remember we said DNA has AGCTs and RNA has AGCs and Us. So this is the A, and that's ribose, so it's an RNA. So RNA has AGCs and Us. It's this one here. And then it has that phosphate group, which we said together in chapter 5. This is called a nucleotide. And if you don't believe me, you can go back and look. chapter 5. And this phosphate has negative charges on it. And then this is another ribose sugar. So essentially, this is two nucleotides. One is A, and another one's weird. So this one, the second one is called N, which stands for nicotinamide. You don't need to know that, but I'm just showing you that these are two nucleotides. This is the base. So this is that N base, that nicotinamide base that's shown here. So it's basically two letters of RNA, two nucleotides of RNA put together, and it has a specific job. So how many of you guys would go stick your finger in an electrical outlet for fun?
Hopefully no one.
I'd say no one too. But the reason is because you don't want electricity flowing around inside your body uncontained. It's the reason that your vacuum cleaner is coated in rubber, right? The cord. What if it was bare? Would you want to be grabbing a hold of that thing?
No.
No. So when we transfer electrons in oxidation-reduction reactions, flowing electrons are electricity. It's the same thing that comes out of your wall. And that would fry you. So you need to have a way to contain these electrons that are getting transmitted throughout your body in a super ordered way, just like an electrical cord would do. And to do that, you need this molecule. You need this NAD molecule. It is an electron transporter. It's like loading up a truck, a UPS truck full of electrons and driving it down the street. You don't want to just shoot the electrons in a big arc down the street. You want to deliver them to where they go. So this molecule has a positive charge on it. This N right here is positively charged. And that's why it's called NAD. It's nicotinamide dinucleotide. That's what the D is. That has this plus charge. So we're this, and that it's dinucleotide because it has two nucleotides. Di means two. All right, so what happens is the glucose from your food has two hydrogens on it. Now let's break this down. So we have two hydrogens, full-on hydrogens. What's hydrogen made out of? Well, hydrogen is made out of one electron and one proton. And there's two of these. So that means we have two electrons and two protons. Everybody follow me? We're going to take this off glucose. Remember, glucose is C6H12O6. So if we split them up, we have two electrons and two protons. Now, we're going to take one of these electrons from glucose and we're going to stick it on this N. So remember, this N was a plus. Now it's neutral. So we're going to subtract one electron. So now we have one electron left and two protons. This went on to the N plus and made it an N. So now we don't have that plus like we did here. It's uncharged. Here's another thing we're going to add is this hydrogen. So you can see that hydrogen is not here. It's over here. And so if we add a whole hydrogen, we need to subtract one electron and one proton, right? So we subtract an electron and one proton. The only thing we have left is one proton, which is shown right here as an H plus. So basically, we're just taking hydrogens and electrons off of glucose and sticking it on NADH. So if the glucose loses an electron, is it oxidized or reduced? Remember, Leo the lion goes grr. So if we lose an electron, we're
Oxidized?
oxidized. So glucose is oxidized And then if we gain an electron, which nicotinamide did, then what's NAD plus now? Is it reduced? Yeah, it's reduced. So this is reduced. This is oxidized. And that's an oxidation-reduction reaction. And the whole point of this is to move electrons. If you got, so it's kind of crazy, but the body has figured out how to use electricity the same way that we have. Like, think about all the things that you own that run off electricity. And if I shut off your power, what you would not be able to do. It's a lot. And your body's been doing this for millions of years. Well, living systems have anyway. And that's what's going on here. We're moving electrons to create electricity, and we're doing it in a safe way. We're moving it from the food we eat to what we call the electron transport chain, which we're going to talk about in a second. All right. So the enzymes that do this are called dehydrogenases. Ends in ASE, so it's automatically an enzyme. What does D mean? If you decant somebody, what are you doing?
Taking it away.
Right. And then hydrogen, right? So a dehydrogenase is an enzyme that takes away hydrogens. And that's what we did, right? We took 2 hydrogens away and put 1 on here, and then electron and hydrogen ion, proton right there. So this is the enzyme that does this to glucose to create NAD+, which is an electron carrier. Right? Remember, its whole job is to safely carry electrons through the cell. We have to do this in baby steps because just so you know, hydrogen and oxygen are things that we use to power rockets that fly to Mars and so on and so forth. The byproduct is water. If you contain so much energy that if we combined your hydrogen and oxygen all at once, you would burst into flames. And a lot of people have argued that this is how spontaneous human combustion occurs, although I haven't really seen much evidence of it, but I guess it's theoretically feasible. But you want to do this in baby steps, so you only release tiny bits of energy as you go along the way, and that way you don't burst into flames. So that's why there's all of these different little steps instead of one big step. All right, so the steps that are involved are called glycolysis. Glyco is sugar, lysis, remember what that word means? Like hydrolysis, it's to break apart. The Krebs cycle, this is also known as the citric acid cycle. The reason is that this guy Krebs figured this out, this cycle. It's not really a cycle, it's just a series of enzyme reactions. Somebody else challenged him because they said they figured it out first. And then a bunch of scientists said, fine, we're not going to call it after any of you guys, we're just going to call it the citric acid cycle. But this name kind of stuck because it was the first name that was used. Citric acid is more commonly used these days. But anyway, that's why they have 2 names for it. And then the last one is the electron transport chain. So we're going to go over each of these individually. On the test, I will ask you where these reactions occur, so make sure you know. So glycolysis occurs in the cytoplasm, in the cytosol. Its job is to convert glucose into a molecule called pyruvate. It's not very fancy. I'll show you a drawing of what it is. It's just a few atoms. And then we make ATP. So this is called substrate level phosphorylation. It's a fancy name for just saying we're adding phosphates to a substrate. And you'll see that whenever I show you the steps in glycolysis. We're going to add phosphates here. We need to do that to break this molecule glucose in half. We make NADH. Remember, NADH's job is to carry electrons, and it's going to deliver those electrons to, fittingly, the electron transport chain. Once pyruvate's made, it goes into the mitochondria. So here's the mitochondria. Remember, it's a membrane-bound organelle. To do that, we're going against the gradient. So if we want to do active transport, what do we have to use? We have to use energy. And what's our energy currency? It's ATP. So we need to pay ATP in here to make this pyruvate go inside the mitochondria. In the mitochondria, in the matrix, right, in the inside part, we talked about this in chapter 6. That's where this Krebs cycle is, and I'll show you what it is. And we're going to make more electron carriers that are going to go to the electron transport chain. And then the electron transport chain itself is also in the mitochondria. This is where so the artist drew this giant explosive ATP. This one's smaller. This is the biggest one. To break it down, we make 2 ATP here. We use the 2 ATP there. So at this point, we have 0 ATP. The Krebs cycle makes 2 more ATP. And then this one makes 34 ATP. So if I were the artist, I'd draw it like way, way bigger, because that's 17 times more ATP that you're making in the electron transport chain. All right, so we're going to take them one at a time. Glycolysis is the first step. So let's just step back and think about what's going on here. So let's say you woke up this morning, you ate your bowl of cereal. Your bowl of cereal is full of carbs. Carbs are amylose, long chains of glucose linked together that plants made, and you put it in your mouth. And your enzyme amylase is going to start breaking those bonds through hydrolysis reactions, and you're going to swallow it. And inside your stomach, you'll end up with maltose. That acid in your stomach is going to break the bonds of maltose into individual glucoses. And, after your stomach is done digesting, it's going to empty its contents into your small intestines, where the glucose is going to be released as well. And since you haven't eaten all night, your blood is going to be real poor in sugar, and your intestines are going to be real high in sugar. And so through passive transport, that sugar will be transported into your bloodstream without any energy required, passively. Once that glucose gets into your bloodstream, it's going to activate insulin in the pancreas. Insulin is a signaling molecule. It's going to bind to a receptor on your cell, and that receptor is going to tell it to build a glucose channel in your cell, and that channel is going to allow glucose to come inside into your cytoplasm. So all of those steps that happened this morning when you ate food is where we're at. So you have glucose in your cytoplasm that came in through a channel that was indirectly built by insulin. And now you have to break down that glucose to release energy. And that's glycolysis. So remember, glucose is a 6-carbon sugar. We want to split it down the middle into 2 3-carbon sugars, which, remember what we call 3-carbon sugars? Do you guys remember?
Try something.
Yeah, sugars is an OSE, so it's a triose. This is a hexose, right? So we're going to cut that in half, and we get 2 trioses. They're oxidized, so what does that mean? Gain or lose electrons?
Loss.
Yeah.
Gain.
Yep, that's right. Remember, Leo the lion goes grr. So lose electrons are oxidized. in glycolysis, we take the electrons off of glucose and put them on the electron carriers, the electron trucks. And then we arrange it into 2 pyruvates. So enzymes do this, right? They're the workers in the factory. This occurs whether or not oxygen is present. So it doesn't matter if you're holding your breath or breathing, glycolysis happens. And there's 2 phases. One is energy investment and one is energy payoff. So in this case, you have to pay to play. And so let's just go through that. So the glucose that you just got from your cereal is now inside your cell. In order to start breaking down that glucose, you have to add 2 phosphates to it. And I'll show you where exactly those occur. But that cost is 2 ATP. Once we put the phosphates on the glucose, we can get 4 add. So it'd be like someone giving you, or you giving someone $2 and they give you 4. You'd do that all day, wouldn't you? And then we're going to make NADH. Remember, these are the electron carriers. And then in the end of this pathway, we're going to have pyruvate. This still has some energy, a lot of energy actually. So we can break it down further in the Krebs cycle, which we're going to do. So in the end, and what I want you guys to know from this lecture is what goes in, and what comes out. So what goes in is glucose and ATP. And what comes out is 2 NADH, 4 ATP, and 2 pyruvate. Right? And that's all you need to know for the test. What goes in and what comes out. And so I'm going to go over this real quick and then I'll end the lecture. You don't need to memorize this. This is what you would memorize in a biochemistry class, but I just want to show you this isn't rocket science or anything special. We have a molecule called a hexokinase. Remember I told you that a kinase is a molecule that adds a phosphate, and then it adds a phosphate to a hexose. Remember, glucose is a hexose sugar. So this enzyme is going to take a phosphate off of ATP and put it on glucose. And we saw this enzyme in chapter 8. It was that big blue molecule that I showed you with the active site. So this becomes glucose 6-phosphate because it's on the number 6 carbon. 1, 2, 3, 4, 5, 5 here and 6 here.
Come here, honey. Come here.
Anyway, I'll just end this here, and then we can pick it up on Thursday. Any questions?
Are there any office hours after this?
Yeah, I'm gonna go get myself a drink and something to eat, and then I'll come back to office hours in a couple minutes. Any other questions?
Nope, thank you.
All right, I'll see you guys Thursday. Remember, I moved all of the due dates out a week, so everything's due a week past what it was before because of the cyber attack on the servers in MCC, the whole district. Anyway, so all the due dates are in Canvas. I haven't updated the syllabus yet, so make sure you follow Canvas and not the syllabus.
Okay.
All right, have a good one.
So
Talk to you later.
when do office hours usually start?
2 o'clock, so I'll be there at about 2:05.
Okay, thank you.
All right.